Thank you, Alina, and good morning and good evening, everyone. And it is my pleasure to talk about the Cytobank job functionality today in today's talk. And today is the last session of the Cytobank Bootcamp's webinar series. And I will talk about, in previous 5 sessions, my colleagues, they talk about how you can use Cytobank to analyzing cytometry data and how you should using different algorithm, machine learning based algorithm to solving your different research or biomarker discovery problems. In today's talk, we will go beyond cytometry data. We will show you how you can apply those machine learning algorithms to analyzing non-cytometry data, including single-cell RNA-seq data, CITE-seq data, and imaging data. So actually, before I start my presentation, I just want to remind everyone again, Cytobank has offer like free 30-day free trial account on Premium Cytobank. If you are interested in trying Cytobank, please feel free to go to the premium.cytobank.org site to sign up and create a new account. All right, let's get started. So to start, you might wonder, what is Job? Job is a data uploader that created by Cytobank developers. And the purpose of Job is to allow you to convert all your non-cytometry tablet data into FCS file format so that you can use the same single-cell analysis tools that are developed for regular cytometry data to analyzing your non-cytometry data. And I think non-cytometry data is really a big word, and it means a lot of different things. But here, we test the job portal with CITE-seq data, single-cell RNA-seq data, and also some of the image feature data. Given the time constraint we had in today, my talk will focus on CITE-seq data and single-cell sequencing data. And for image data, it's like I will briefly describe the workflow for the image data, but it will very similar to what you will do or what you will encounter when you're analyzing like single-cell sequencing data. So actually, Job is like I mentioned in previous slides, it's a data uploader, and you can upload your text format, like usually normalize and filter the data into Cytobank through Job. But besides Job, there are many other important steps are involved in your single-cell RNA-seq data analysis workflow or your CITE-seq data analysis workflow. In order to help you to get a big picture, a better understanding of the complete workflow, I would like to briefly discuss this like 3-step workflow that we proposed to help you get a better idea about kind of give you an overview about what is the best practice or what you usually will need to do with your single-cell or CITE-seq data. So in a nutshell, like single-cell RNA-seq and CITE-seq data, they are very similar, and they all use like very often, like you will generate data using Illumina sequencer, and these two technology are all like next generation sequencing based technologies, and you will getting digital signals rather than analog signals from your data. But for to start, what you usually get from this after you perform the actual sequencing experiment is the raw sequencing rates. And if you have a CITE-seq data set, you will get 2 different kinds of expression information. One is the single-cell RNA-seq information, and the other one is the single-cell protein expression information. And if you only if your data is a single-cell RNA-seq data, you will only get the RNA-seq expression informations. You will not get the protein expression informations. And then you can take your raw sequencing rates, go through this 3-step process to get your biology insight. And in step 1, you will need to map those raw sequencing rates to a reference genome, and then generating the raw expression count with both your RNA-seq and protein expression matrix. And particularly, I list the possible tools that you can use in this step. So for example, you can use the 10x genome cell ranger tools or the star aligners to map your rates to the reference genome. And then both cell ranger and star can automatically generating RNA-seq expression count for you. If you have a CITE-seq data, you will need to use the CITE-seq count, which is a Python script that will generating the protein expression count data for you. After step 1, you will get some raw expression count matrix, and the data are usually stored in a text file. And then you can take the text file to start the step 2. In step 2, the main purpose of step 2 is to normalize, filtering, and QC your data. It's similar to what you will do for your cytometry data. It's similar to the cleanup gating process of your cytometry data, but it is much more complex than that. So I highlight some of the key steps in step 2 here, but we will go into details later. Here, basically, you will filtering the data, normalize the data, and then at the end, you want to leverage the cell barcode information to combine, to merge your gene and antibody expression into one big expression matrix. So after step 2, again, you will have this nice filtered and normalized and very often scaled expression matrix. And now is the time you can use job to import and process the data into Cytobank. And as soon as the data is in Cytobank, you can do many different kinds of analysis in Cytobank. So for example, you can run Visney to visualize the underlying data pattern of your CITE-seq data or single-cell RNA-seq data. And then you also can run flow-sound to identify cell clusters or use citrus to do biomarker discovery. And from there, you will help you to you can go to get your biology insight from your data. And for image data, the workflow will be different. Actually, it's more simple. For image data, you will need to use some third-party traditional image analysis software to get image feature data. So for example, you can use the cell profiler, which is developed by the Border Institution, I believe, and you can use the software to generating image features such as like the length of the cell and the size of the cell and different kind of features. And then you can import that numerical feature data set into Cytobank using job. So you basically can skip the step 1 and step 2 showing in this RNA-seq workflow. And for image data, you will only need to use cell profiler to generate the feature data, and then you can import the data into Cytobank. So next, I would like to kind of go into a little bit more details to discuss all the 3 steps that we proposed for this CITE-seq workflow. So again, in step 1, the main purpose of this step is to map the raw sequencing rates to the reference genome and then generating expression count. One thing I would like to mention that CITE-seq data contain 2 parts of expression information, the RNA-seq and protein. And for single-cell RNA-seq data, it will only contain the RNA-seq expression data. And regardless what kind of a single-cell sequencing data do you have, you need to map those raw rates to the reference genome. And then here's I have some practical advice for you. So if you want to map the raw rates to reference genome by yourself on your local computer, you probably need to get a very powerful computer which has to have at least 30 GB of memories to analyzing the human data. And then I also suggest you need to download the pre-built reference genome files from 10x website or from the Illumina iGenome project website, so that you will not make any mistake, like you will not getting any misleading results by mixing different reference genome file during your alignment process. And then again, you will use the CITE-seq Python script to process the protein file. And then in fact, the input file for the CITE-seq Python script is a BAM file. BAM file contains the aligned read informations, and FASTQ file is the raw file that has the raw sequencing information. And again, actually this step is, I recommend you, if you are not familiar with programming, you should try to get some help from your bioinformaticians. If you cannot easily find bioinformaticians, Cytobank, we also have the data consulting service team, which I'm part of it. And we can help you to process and analyzing the data for you. And just in real world, very often bioinformaticians, they would not like do the map alignment process on like small desktop machine, and they will usually use Linux clusters or open a virtual machine on AWS or Google Cloud to use the big virtual machine to process this data set. And next, in step 2, after you finish the alignment and generation and generated the raw expression count, you can go ahead to pre-processing those raw expression count. And I would like to mention that this is step 2 is really important, because it will help you to build the step 2 will help you get a clean copy of your data for all your downstream analysis. And here, like you need to do several different filtering. Actually, if you can imagine right now, the data matrix you will get from after step 1 are a n by m matrix. So the rows are the genes or features, and the columns are the cells. Because this data matrix has 2 dimensions, when you filtering your data, you are not only need to filter on the cell dimension, you will also need to doing filtering on the row dimension, which you will need to filter in genes based on some criteria. And this is also different from the regular cytometry data analysis, which you would not like kind of remove channels or remove markers unless you need to merge data that are generating on different panels. But here, for CITE-seq single-cell data and single-cell RNA-seq data, it's quite common that you probably will remove genes or to get a clean copy of the data for the downstream unsupervised learning analysis. And the other thing I would like to mention that is really important for you to do some normalization, because again, the sequencing data is different from the cytometry data. And during the sequencing process, you will get a library coverage bias. And this is a known technical factors or bias that you need to deal with. And so at minimum, you have to do normalizations to remove that library size bias for your RNA-seq data. And then at the end, very important, in order to analyze the data in Cytobank, you also need to combine the RNA-seq and surface protein expression data by using the cell barcode provided in the raw expression data matrix. And I also would like to mention, if you happen to pull multiple samples together and sequence the pooled samples in one sequencer lane. You probably need to demultiplex your data at the end of the step 2 so that you will get one normalized and filtered expression matrix per sample. All right, and after the very complex step 1 and step 2, now you are in step 3. This step actually is really important. It's really easy comparing to the first 2 steps. Here, you just need to use the job in Cytobank to upload your data into Cytobank, and then you can run all the Cytobank machine learning tools with your CITE-seq data or single-cell RNA-seq data. Later I will do a live demo to show you how can you import the data using job in Cytobank. And specifically, I would like to mention that in step 2, we provide a Cytobank R script. Again, if you can get help from bioinformaticians, and they probably have their own customized script, and you can trust them and just like let them to do the filtering or normalization for you. But if you are interested in doing this, the step 2 for yourself, and you are familiar with R programming, and you can download the Cytobank R script and use the script to process your raw expression data. And the script can read data from the MEX file format. It also can support the CSV files. And here I would like to mention that if you are enterprise Cytobank customers, you can import as many as about 800 features into Cytobank. And if you are a premium Cytobank users, you are only allowed to import as many as about 100 features into Cytobank. And there are some workaround for you to deal with the feature limits for premium user. One thing you can do is actually in step 2, you can add in one more step, which is running PCA principal analysis to reduce the dimension of your gene expression matrix into a lower dimension. So for example, your raw data has maybe 5,000 genes, high quality genes, and then you can run PCA to create meta genes, which will contain a combination of gene expressions from those 5,000 input genes. And then after PCA, you can select maybe the first 20 principal components, and then use that as a feature and then import those features into Cytobank for your downstream analysis. And I also would like to mention there's no limit for number of cells you can import regardless you are an enterprise or premium Cytobank user. All right, so next I will show you an example with real-world CITE-seq data and tell you how you can use Cytobank to analyzing the public data set. So the public data set that I will use in my today's talk is a core blood CITE-seq data set. The data is downloaded from the Satija lab website, and the data has about 9,000 cells and 36,000 genes. It is a CITE-seq data set, so it also has 13 protein markers, including some of the popular markers such as CD3, CD4, CD8, CD19, and so on. And when you download the data from the website, actually they already help you to finish the step 1, which is map the raw expression counts to the reference genomes and then generate the raw expression count matrix for you. And you might wonder, how does those raw expression count matrix look like? And here I provide 2 screenshots for you. So if you look at the pictures at the right, This is the screenshot of the raw protein expression matrix. As you can see, the rows are the CD markers, and we have like 13 different antibodies in this data set, and then the columns are the cells. And the header, the column header are the cell barcodes. These are the unique barcodes, and then we can use these informations to combine to kind of link the RNA-seq expression informations with the protein expression informations. And the other thing you might notice is that if you look at these values, they are all integers. This is one difference between the sequencing technology and the cytometry technology. Cytometry technology, for cytometry technology, you will get real number, which you get like kind of analog informations from by reading the fluorescent intensity from during the data acquisition process. But for sequencing data, you will getting digital count, and you will getting integer numbers. And right now, if you look at this, like the screenshot at the left here, this is the screenshot of the raw gene expression matrix. Again, the rows are the genes, and the columns are the cells. And you might also notice that we see loss of a zero in the screenshot. And actually, this is quite common, and this is a common issue with single-cell RNA-seq data or CITE-seq data, or the RNA-seq part of the CITE-seq data. And because the sequencing technology can allow you to simultaneously measure maybe like 20,000, 30,000 different features. So very often for a particular cell, you would not see all the genes are expressed at the same time. Very often, only a small subset of genes will express some signals in the real biology environment. And that's the reason why you see so many genes that has zero expression count across all the cell. And that is the reason, and that's the reason, one of the reasons why you need to doing different kind of gene filtering to remove these low expression genes. If you don't remove these low expression genes, when you apply normalization to correct the library size effect, you will introduce noise or bias into the normalization process, and then you will end up getting a low quality normalized data. And so right now, and after you finish run, assume you run Cytobank R script, or your bioinformaticians help you to filtering and normalize your data, you will getting a big expression count matrix. And here again, this is a screenshot of the matrix. And one thing I would like to point out is that this screenshot will look different from the previous two, because here we transport the matrix. In this normalized and filtered matrix, the rows are the cells, and the columns are the features, so including RNA-seq and protein for CITE data. And this process, the matrix, is similar to the regular cytometry data matrix, because in cytometry data, you usually have rows as cells and columns as markers or antibodies. And next, you can import the process and normalize the data into Cytobank and run, for example, the first thing you can do, you can run Whitney. And Whitney, again, is an unsupervised machine learning tool, which allow you to study the underlying data pattern of your data. And in this particular example, we have the CITE data set, and we can run like, there are many different ways to run, to apply Whitney with your CITE data. Here, particularly, I show you can run Whitney with only your with only the RNA-seq expression matrix. You kind of don't use the protein expression matrix for your reasoning analysis. And then you will save that protein expression matrix information, and you can, based on the protein expression, to identify or phenotype the Whitney cell clusters after you complete the Whitney with your gene expression data. And this screen shows you the result of this analysis. As you can see, the Whitney can separate cell events based on RNA-seq expression data. We got pretty nice separations on these Whitney results. And then when we overlay the protein expression data on this Whitney map, we find that like we have B cell islands here, we have a large CD4 positive T cell islands, and then we also have a small CD8 positive T cell islands in this data. So you can generate this very nice graphic in Cytobank and to show the results to your collaborators or colleagues. And you might wonder, like, how can you generating these kind of results in Cytobank? So I briefly kind of show you the workflow to get those pictures. So the first thing you need to do, first of all, you want to set up a Whitney run in Cytobank with your RNA-seq expression data. And then at the end of the Whitney run, you will get this black Whitney map, which is showing at the left. And then you can overlay different antibody expressions on this Whitney map. So for example, here, if you look at the 4 plots at the middle, we overlay 4 different antibody expressions on this Whitney map. So if you look at the CD19 map, you will see here we got one island which really has very high CD19 expressions. So we know that this is a B cell. And then you can go to the gating page in Cytobank to draw gates based on the overlaid antibody expression Whitney map. So here again, show you an example. You can draw different gates in Cytobank and define all these populations. And since CITE has both the RNA-seq expression data and antibody expression data, very often people will ask or wonder, how well does the protein expression correlate with the gene expression? And there are many ways you can to analyzing your data and answer the question. But here particularly, I want to show you one example, which you will use a heat map in Cytobank to inspect the relationship between the protein expression and the corresponding protein coding gene expressions. So here we create this heat map. Again, the rows are different cell populations, like annotated cell different populations, and the columns are different markers. So including protein coding genes and the regular antibody. So if you look at the first 2 columns, you will see the column number 1 and 2. These 2 are the protein coding genes, and column number 3 is the antibody expression. And if you look at the heat map, you will see this, and they have exactly the same expression patterns as showing on the heat map. So you know that for this particular CITE data, the protein expression is well correlated with the gene expression. And in Cytobank, you also can create the overlaid histogram to visualizing the surface marker expressions. And this is similar to what you will usually do for your cytometry data. Again, in these examples, we show 3 different populations in this overlaid histogram, and different columns are the expressions of different antibodies. And if you look at, again, see the CD19, you can see this blue populations, which is a B cell, has extremely high expressions comparing to the T cell populations in CD19. And then the first two analysis is kind of you apply different machine learning algorithm with one of the either like RNA-seq or protein expression data first, and then you kind of using the other piece of the information to validate your results. Here, you also can do some kind of an integrated analysis with your CITE-seq data. And suppose you want to assess differential expression of genes on B cells across different samples. And what you might can do in Cytobank is that you can use this integrated analysis approach to analyzing your data. And I would like to point out that for this particular proposed integrated analysis, you need to have multiple samples. It cannot be performed with one sample. And this is a two-step process. So in step one, you will run flow sum in Cytobank to identify B cell clusters using the CITE-seq protein expression data. And then you can look at the differentially expressed genes based on full change values as soon as you know which cell clusters are the B cell cluster. So again, this is on kind of a purely machine learning based approach to analyzing, to seeking answers for the proposed question. In step one, you will run flow sum with your protein expression data. And here in this particular examples, I set up a flow sum run and to generating 8 different meta clusters. So as you can see in this heat map, we will have 8 different rows in this heat map, and the columns are the expressions of different antibodies. And particularly, if you look at the meta cluster number 1 and look at these CD19 expression columns, you will notice that this meta cluster 1 is likely to be a B cell cluster because it has really high CD19 expressions. And now we know that meta cluster 1 is a B cell population. We can move on, go to step number 2. And now you want to comparing gene expressions on B cells between different samples. So if we have 2 samples in this particular data set, you can generate a heat map. Again, the columns are the samples and the rows are the different genes. And then we will color code this heat map based on log fold change values. And you can get log fold change values easily in Cytobank by using the Cytobank statistical table function. As you can see, here we got some genes that is upregulated in sample 2 comparing to sample 1, such as GPX4, and then some of the genes are downregulated, such as TCL1A. So this is a very simple, nice example that tells you how you can do some integrated analysis to analyzing a CITE-seq data. And I would like to mention that the CITE-seq technology is a new technology, and there's no golden standard process in terms of how you should analyzing your CITE-seq data. So, and the several examples I provided here, it is just kind of show you some of the possible options that we provided for you in Cytobank, and hope these examples are useful. And if you have more questions, and I would like to point you to the blog poster, and also we also have a YouTube video and to show you how you can analyzing the CITE-seq data and in Cytobank. Now I will do a live demo to show you how you can actually import the data into Cytobank. So let me share my screen. The first thing I would like to actually, I want to kind of start from the right beginning of the workflow, which kind of show you how a FASTQ file might look like. I guess some of you might be interested in knowing that. So here, this is some of the FASTQ file I had on my computer. As you can see, we have several FASTQ files in here for this particular data set, and the size of these files are huge. On average, they're about like 10 gigabyte per file. And some of you might notice that we have three different group of file. We have the ADP FASTQ file, HTO FASTQ file, and RNA-seq FASTQ file. And the reason why you get three different kinds of FASTQ file for this data set, because in this data set, we put samples together to when we do the sequencing, and then and the antibody is the ADP FASTQ file contain the antibody expression informations, and the RNA-seq FASTQ file contain the raw RNA-seq rates. And next, I can just quickly open one of the FASTQ file and show you how it will look like. And this FASTQ file, again, is really big. You don't want to use a text editor to open it. And what you can do, you can use the, like on Mac, you can use the terminal tool, use some of the command line program to view the content of this FASTQ file. Here, I open one of the FASTQ file. As you can see, we have multiple lines here. And every 4 lines representing one raw sequencing base. And the first line are usually our sequence race unique identifier. And the second line is the sequence. And then we have this plus sign as a separator. After the separator, we have a line of quality scores. Basically, the quality score tell you for each base pair and how likely you the sequencer make a mistake by calling this position as A or T or C or G. And so this file is really big. Again, you want to find your bioinformatician to help you to analyzing it. But if you are interested in learning how to do it, and there are many online education resources available, you can Google it and find out. And next, I would like to show you. So you will get how you can import the data into Cytobank. So for example, you download these 2 raw expression matrix from the public website, and then you kind of run the Cytobank R script to filtering and normalize your data. Now you can, after the filtering normalization, you will get one normalized gene expression count matrix CSV file. This file contains the normalized data, and now you can import this data into Cytobank. So let me open my Cytobank page and show you how you can do that. So to start, you want to create a new experiment. So give a name of the experiment, job, and then you can just select the file and drop it to here and stop the uploading. And you can call it, this is CITE data. All right, I want to stop here for a moment, and this is really critical step. Very often we get a lot of questions from users about which cells should they select. So the rule of thumb is that you need to select the first numerical cells in your data matrix. So for example, in this particular data examples, as you can see, the first numerical like number cells is the cell in row number 2 and column number 2. You don't want to select like the column number 1 and row number 2 cell, and you should select the cells in row number 2 and column number 2. All right, after you make your selection, you need to confirm it with Cytobank. Now the Cytobank start to upload the data into the cloud server, and depending on your home internet speed, it could take maybe, like sometimes if you have some large data set, it could take a very long time, maybe up to 1 hour. But if you have a relatively small data set, it will only take maybe 2 or 3 minutes. And today I'm using my home internet to do the webinar and do the live demo. So it's a little bit slow for me because I'm streaming the webinar at the same time. I'm also uploading this data file to the server. So I already pre-built another experiment yesterday, so we can continue the demo with this pre-built experiment. You upload the data in Cytobank. As you can see, you will get one for this particular example, you will get one file. And then the first thing you should do, you need to go to the data scaling page. And it's really important for you to confirm the scaling settings for your data, especially for non-cytometry data. My recommendation is that you should apply linear scale for your non-cytometry data, because very often you will do normalization and filtering and data standardization outside of Cytobank. Here, like you don't want to like reapply a transformation on your data, and so I would not recommend you using log transformation or R-side transformation unless you fully understand the data distribution of your data. But you are welcome to like change the settings and play around it to see which one can give you the best result. So after you confirm your data scaling settings, and you can go ahead to start a Whitney analysis like I show. So let me show you how you can create a set up a Whitney analysis. And the first thing you need to do, again, you need to select one of the population. In this particular example, we will use all the cell events in this file and to run this Whitney. So as you can see, we have about 9,000 cells in these examples. We will use all the events. And here, this is the interesting part. As you can see, in this particular data set, we have 700 channels, which is huge. I think for cytometry data, you will get maybe like 20, 40 channels for flow data, and then you will get maybe 80, 90 channels for CITE-seq data. But for CITE-seq or single-cell RNA-seq data, you can get like a lot of channels. And then you want to, in this example, I want to run Whitney with only genes, not the protein expressions that are generating in the CITE-seq, contained in the CITE-seq data. So I will select all the channels, and then I will deselect those antibodies channels. So I search for CITE-seq and just unselect all the antibody channels. Now, and as you can see, we will run this Whitney with 700 channels, and then you definitely don't want to do uncompensation because there's no compensation data available in this non-cytometry data. And in order to make sure you can reproduce your Whitney analysis result, I usually like to set a C. and then you can leave the rest of parameters as the default value. Now you are ready to hit this big green button to start your busy analysis. All right, so I would like to stop here and open the question to make myself available, and also my colleagues to answer any questions that you might have.
Thank you so much, Jihad. This was great. A live in-app demo and a really good presentation on how to use the algorithms for non-flow data. So first and foremost, I'm going to introduce everybody. So everybody on the panel. So we've got Jihao, our data scientist. Steph Troff is with us as well. She's an additional data scientist. We have Chin Jun, who is our application scientist, and and Sarah, who is our sales specialist for the East Coast, and also Richard, who is the sales specialist for the West Coast. So there's a lot of individuals here at Cytobank who are going to be able to help you answer the questions. And once again, I should also say that my name is Alina Lelich, and I apologize, I always forget that part. So you guys saw that we just wanted to advise you again that you can try Cytobank for 30 days for free. And please do download it off of our website. Chihao has provided you the link. In addition, I also wanted to let you all know that there's going to be a webinar by Dr. Sharam Kordasti, who has actually utilized Cytobank platform to analyze and phenotype Tregs in health and disease. So he's going to be talking about the lessons from the bone marrow failure syndromes and discussing his utility of the Cytobank platform within the data analysis that he's conducted. So please do sign up. It's a Lab Roots webinar. It's going to occur on May 12th at 8 a.m. Pacific time. So if any of you want the link to this, I can post it in the chat. So without further ado, we're going to try to tackle these questions and answer them for you guys. All right. So the first question that we have is, can we use the Surrott algorithm to do the pre-processing step 1 and 2? And I'm going to refer this question to Chihao.
Yeah, sure. Thanks, Alina. And yeah, the answer is yes. In fact, we also create a Cytobank R script based on the Surrott package or Surrott algorithm. And so, yes, in short, you can use it. But again, like I mentioned, there's no golden standard in right now for how you should analyzing your CITE-seq data and single-cell RNA-seq data. And so I expect to see more and more new algorithms coming up, which will provide maybe better results for your analysis.
Perfect. Thank you very much. Okay. Another very important question. Will premium license of Cytobank support this analysis? So for example, started from the FASTQ needed storage commitment, or what will be the working with it after the cell range processing? I hope, so Jihad, do you understand the question, or do I need to decode it a little further?
I can see the question, and I believe I get it. So first of all, for like the raw sequencing realignment, Cytobank doesn't support that. So you cannot basically upload FASTQ file to Cytobank, and sorry, you cannot do that. And you should just get help from a bioinformatician, or you can use the Cytobank consulting data analysis consulting service to help you to process your FASTQ file. But after you run Cell Ranger with your FASTQ file, you will get this raw feature BC matrix file. And if you're saying you don't need to analyze and normalize the data, or you don't feel comfortable to do any filtering, and you can directly upload it to Cytobank and analyzing it. And that also is possible. But I recommend that you run the Cytobank normalization and filtering R script and to process your data and then upload the processed data into Cytobank.
Perfect, thank you, Jihao. Another question, is Drop available to download on my computer? And I'm gonna get Steph to answer this question.
Hi, so Drop is part of the Cytobank platform that you can access by visiting the website and logging into your account. There's nothing that you can or should download in order to use it.
Excellent, thank you very much. Okay, so I think I believe that one of the participants would like to have the explanation for the normalization and the process for it. And if you can just summarize that again, Jihao, that would be great.
Yeah, this is a great question. And actually the normalization process is very complex and also depend on what kind of information you will have. So if let's take the most complex example. So for example, your CITE data has the cell hashing antibody information in addition to the regular surface antibody expression and RNA-seq matrix informations. And for this kind of a data set, the first thing you need to do is like you want to remove liver size bias with the cell hashing antibody expression matrix, and then leverage that information to identify doublets and dibbers. And then after that step, you will know which cell are doublets, which cell are dibbers, and you will apply that to your RNA-seq expression matrix and your antibody expression matrix. And you kind of finished like the cell filtering step at this point. The next thing you would like to do is you want to filtering and normalizing your RNA-seq expression matrix. So the first thing you should do, you should remove genes that has very low expressions from your data, and so you can like remove. You can apply a filter to remove those genes, and then you also need to remove cells that has very low gene expressions. And again, you do all this kind of filtering and normalization with your RNA-seq expression matrix, and then, but since you will remove some cells based on RNA-seq expression matrix, you will also want to remove the same cell in your antibody and cell hashing antibody expression matrix. So after you finish filtering and normalization with your RNA-seq expression matrix, you should apply the same cell filtering informations to the other two antibody expression informations. And so basically at the very beginning, and just to summarize, at the very beginning, you need to deal with your cell hashing antibody expression matrix, and then you move to filtering and normalizing RNA-seq expression matrix, and then at the end, you want to deal with your regular antibody expression matrix. So, and then at the end, after you finish all the first three steps, you want to combine the RNA-seq and regular surface antibody expression matrix into one big matrix. And at that point, you can choose to do PC analysis with that filtered and normalized data. Or if you are enterprise user, you don't need to do that. You can like just select the most variable features based on that normalized and then filter the gene expression counts, and then import the data into Cytobank using Drop. Yeah. But if you are interested, I can send you an email later to kind of outline the detail about what kind of a normalization and filtering you need to do for your CITE data.
That's great. Thank you, Jihao. And also with that, I just want to remind all of our attendees that we do have, there is a technical support portal that you can write into, and we will send you the link within the chat as well for that. So thank you. Okay. A really good question as well. Can we transfer metabolomic or microbiomic data into Cytobank? And I will get, Jihao, did you want to tackle this one again?
Yeah, sure. Um, Actually, I'm not familiar with the microbiome, these two kind of data, but as long as the data is a numerical data matrix, you can import it into a Cytobank. And the one thing you need to pay attention is that because in Cytobank you will run unsupervised analysis, you should always make sure that you will standardize your input data so that like features that are on different scales or features that has different mean values can be run together in one unsupervised machine learning analysis.
Perfect. Okay, so this is a more of a data set. So this is from an attendee who says, I have patient data that is on facts and so flow cytometry data and non-flow cytometry data. Should these two data sets be integrated into one table for patient IDs and as columns and parameters as rows?
Yeah, thanks.
Parameters as rows, yes.
Yeah. Yeah, thanks for the question. I assume you want to analyzing both the both data set in Cytobank. So for that purpose, you want to make a matrix that columns are the features or parameters and the rows are the patient IDs or cells of a particular patient.
Okay. All right, so this is, how do we integrate patient FCS data with corresponding, so this I guess is, it's similar to the question above. So how exactly do we integrate flow data with non-flow data such as Luminix in DROP? Does it need specific transformation or normalization across the facts and the non-facts before integration or before the running of the algorithm?
Actually, first of all, you need to combine these two type of data set outside of Cytobank. Unfortunately, Cytobank doesn't not allow you to like combine the flow data and non-flow data together like easily. If you want to like but one thing you can do, you can kind of create in the separate data file for this information. So for example, you will create one file that contain only the flow data and the other file contain the no flow data, but the data are generated from the same patient. But again, it's really depends on what kind of analysis you want to do with the data. So without having any more information about that, it's hard to provide any suggestion for your question. And just in terms of data transformation and normalization, again, for flow data, you just need to do the regular data transformation. And when you combine these two data, different data type together, for sure you need to standardize your data or doing the data scaling or turn on the data scaling functions in Cytobank before you're running any unsupervised learning analysis tools, because you want to make sure that like antibody channels generated in the flow data can be compared with gene expressions that are obtained from your single-cell RNA-seq data. Without doing a data standardization, you will get misleading results from your with-me or flow cell analysis.
Thank you so much. And I'm just going to tackle one more question because so that way we can answer it, so that we can answer all of them. And this is a question that asks, does the premium access have, oh, sorry guys. I've just had some technical difficulties that said that I'm logged out, so never mind. Do we have access to the scientific processing assistance with the premium access level, or is that an additional fee? Jihao, did you want to answer this one as well?
Yes. Yeah. Thanks for the question. There is a fee for using the Cytobank consulting service. So Cytobank scientific consulting service is a paid analysis service. And if you are interested, please feel free to send us an email, and you can find us on the Cytobank main website. If you go to service, scientific service, and you can provide some information about what kind of analysis you are interested in, we can set up a 30-minute free consultation with you to create a strategy for you.
Perfect. Okay. So that concludes our seminar for today. Thank you all so very much for attending and joining us for all the 6 sessions, or the particular ones that you found of interest. It's been our pleasure to help you stay home and get educated during this time of physical distancing. Stay healthy and stay safe. And thank you once again for all of your attendance. And don't forget, there's that Lab Roots webinar that's going to happen on May 12th. So please write to us. Let us know if you have any further questions. And thank you all once again.
Thank you, everyone. Bye.